To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.
Hello my dear students, welcome to today's lesson. So I am going to continue with the lesson Inheritance. In the previous chapter, we discussed about Gregor Mendel, father of genetics, and he did experiments related to inheritance. So actually, he did the experiments and explained inheritance through his observations. And you all know he used the garden pea plant, that is Pisum sativum, for his experiments, and we discussed the reasons why he selected that particular plant. And the experiments and his observations are related to monohybrid cross. You all know the meaning of monohybrid cross. Mono means one. He considered a single pair of contrasting characteristics. For example, we discussed tall and short plants. Similarly, he observed 7 different characteristics of the garden pea plant or the Pisum sativum plant. And from all those, he used the parent generations, which were pure breed plants. Then the F1 generation was produced where only the dominant character was observed. There was the recessive character that was not expressed in the first generation. Then thereafter, he observed the second generation where again both the dominant and recessive characters were observed and in the ratio of 3:1. And you can remember there was an assignment where you all were supposed to analyze the scientific method that was used by Gregor Mendel to carry out his experiment. So based on that, now we will try to understand what inheritance is based on Gregor Mendel's experiment and his observations. So the inherited pattern of a monohybrid cross. So already you all know the meaning of monohybrid cross. One pair of contrasting characteristics. The results obtained from the Mendel monohybrid cross using 7 different contrasting characters are given in the table below. So we have the table in the next slide, but you all have an idea already. So I told you all we use the tall and short plants. Tall and short plants. And I told you all when you get the F2 generation, we get a 3:1 ratio. That was Mendel's observation. But then again, I explained to you all, he used a large number of plants. So it will not be exactly 300:100. It can be 298:102. But still, if you take the ratio, it will be 3:1. So that is why Mendel carried out the experiment with large number of plants, and also he repeated the experiment to make sure that all the time he got the ratio 3:1. And out of these 2 characters, you can remember the short character was not observed in the first generation. So based on that, he was able to explain that the tall character is the dominant character and the short character is the recessive character. So those were the information we obtained from the monohybrid cross. Now I'll move on to the next slide where we will look at the observations he obtained. So the results of the experiments of Mendel. This is given in your textbook, students. There is a typing error, experiment, EXPERIMENTS, the results of the experiments of Mendel. So in the first column, we have the character. Then second column shows how the cross breeding was taken place. Then F1 generation, what will be observed. So I will fill that for you all. Then F2 generation, we have the dominant and recessive characters. And then the closest ratio. So like I told you all, you won't get an exact ratio, but you will get a very close ratio. So that is how we find the information, observation and the conclusion obtained from the observation. So the first set of characters, color of flower. Now you remember monohybrid cross. So one character is the color of flower. In that there are two contrasting characters. The flowers are purple and white. So color of flower, one feature. In that two contrasting characters, purple and white color flowers. So what do you think? F1 generation. Now if this is not given to you, now you can't just guess which is the F1 generation. It was actually Mendel's observation. What he observed was the purple color flowers were the only flowers that were produced by plants of the F1 generation. But if you are given a table like that, by looking at these two values, you can identify which is dominant. If it is dominant, that will be in the 3 ratio. 3:1. Dominant is to recessive, 3:1. So, no more number of plants having that particular character means that is the dominant character. So, here you can see in the F2 generation, purple is 705, white flowers 224. So, that means purple flowers were produced by more number of plants. So, that means purple flower is the dominant character. So, color of the flower, purple and white. Out of that, purple flowers. All the flowers will be purple. Then here, dominant character is 705, purple 705, white 224. Now, you can get the ratio. You can check it. Now, if I multiply, since we already know the ratio, normally what you are supposed to do is you are supposed to divide 705 by 224. But here we know already the ratio is 3:1. So to confirm, if we multiply 224 by 3, you can either divide 705 by 224 or multiply 224 by 3, you will get it as 672, whereas the number observed is 705. So from that you can understand it is a ratio of 3:1. Or even if you divide 705 divided by 224, you will get a value a little more than 3 point something. So, but we take the closest ratio. Closest ratio is going to be 3:1. It will be 3:1. So, even if we divide it by 705 divided by 224. So, you all know 200, 3 times it's going to be 600. So, you can have it as 3 times, 3 into 4, 2, 7, 6. We have already calculated 672. So, if you subtract 3, 0. So, 33. Then, if you put a decimal here, 330, it will be 1 times 224, that is going to be 116. So, if it is 1160, it could be about 5 times. So, 0, 11, 11. So, 1110, so that's 50. So, it is about 3.15. So, it is going to be 3.15:1. But we can get the closest ratio as 3:1. The same way, if you take the second set of observations, 6022, 2001. So, from there also you can easily see 2000 and 6000. 6000 is 3 times 2000. So, closest ratio is going to be 3:1. So, if you want, you all can divide and check. But that is not really necessary because we already understood from the Mendel's experiment all the ratios were close to 3:1. So then, if we look at the second set of characters, color of the seed, yellow and green. So the contrasting characteristics are yellow and green, color of seed. And out of that, yellow is the dominant character, because here you can see yellow 6022, green 2001. So the closest ratio is 3:1. Then shape of the seed, round and wrinkle seed. Round is exactly round like this. It will be a round seed like that, whereas wrinkle seed, it is going to be something like that. The edges are all kind of shrunk in wrinkle seed, round and wrinkle seed. So there you will see round and wrinkle 5474 is to 1850. Now you can see he has used about 8000 plants. 5000, 2000, 2000, so it's almost 7500 to 8000 plants. A large number of plants were used. So this also, the seeds will be round. Round is the dominant character. So round is to wrinkle. So all the first generation will be round seeds only. Wrinkle seed is not expressed in the first generation. In the second generation, round is to wrinkled. The ratio is 3:1. Then the shape of the pod. You all saw what the pod is. Pod is the one that contains the seeds. Inflated, constricted. You know the meaning of inflated and constricted. Inflated. Now balloon, when you blow air into the balloon, it inflates. So it's a full pod. Whereas constricted, the pod is somewhat shrunk like. So inflated and constricted pod. So in that also, inflated character is the dominant character. And here again you can see another pattern. When we write the characters, normally the dominant character is written first. Dominant, recessive, dominant, recessive. Round is dominant, wrinkled is recessive. Inflated is dominant, constricted is recessive. So inflated 882, constricted is 299. If you get the ratio, that also will give you a value of 3:1. Then color of the pod. Again green and yellow, contrasting characters. So from this you can understand green is the dominant character. So all the F1 generation plants will be green in color. They will have green color pod. F2 generation, green is to yellow 428, yellow 152. So green is to yellow will be 3:1. Position of flower, whether it's an axial flower or the terminal flower. So their axial flower is the dominant character. Axial flower in the second generation 652, terminal flower in the F2 generation 207. So axial is to terminal, the ratio is 3:1. Then the seventh character, the last character, height of the plant, which we discussed first, tall and short. You all know the tall character is dominant, so all the F1 generation is only tall plants. Tall 787, short 277. So here also the ratio is 3:1. So here students, you don't need to memorize these values. You just need to understand it. But you have to know what the dominant character is, recessive character is. So you will have to understand that all the F1 generation will only have the dominant character being expressed. So out of all these, when you say color of flower, purple is the dominant character. Color of seed, yellow is the dominant character. Shape of the seed, round seeds are dominant. Shape of the pod, inflated pods are dominant. Color of the pod, green pods are dominant. Then position of the flower, axial flowers are dominant. And height of the plant, tall plants are dominant. Only the dominant character is expressed in the first generation. But in the second generation, you will get a ratio. And always the ratio is 3:1. Closest ratio. Earlier also I told you all it would not be an exact ratio, but you will get a close ratio. So dominant and recessive characters, you will get the closest ratio. Purple is to white flowers, 3:1. Yellow is to green seeds, 3:1. Round is to wrinkle seeds, 3:1. Inflated is to constricted pods, again 3:1. Green is to yellow pods, that is also 3:1. Axial is to terminal flowers, that is 3:1. And tall is to short plants, 3:1. 7 different characters, and each of them having contrasting characters. So that is the observation or the result of the Mendel experiment. From all these only, he proposed the concept of inheritance. He explained what inheritance is. And remember again, students, he did not have the knowledge about genetics, genes, or even about DNA molecules. That information, that knowledge was not there when Gregor Mendel explained inheritance. But whatever he explained is the correct explanation for inheritance. So we will move on to the next slide. It is clear that all above characters are inherited in the same manner. That is clear, no? They are all inherited in the same manner. Always dominant character is expressed in the first generation. Then in the second generation, it was a 3:1 ratio between dominant and recessive character. So all are inherited in the same manner. One feature is completely hidden in F1 generation and it appears in F2 generation. That is what we call as recessive feature or recessive character. Short character, white color flowers, terminal flowers, all those are recessive characters. Then in both generations, all the plants were with the features of parent generation. No pea plants were with intermediate characters. What does that mean? Now we had tall plants, short plants. We did not have any medium height plants. So the characters were not intermediate, either tall or short. So parents were tall and short. First generation only tall, second generation again tall and short. No intermediate height. Same goes for white and purple flowers. Parents have white and purple flowers. You get purple flowers in the first generation. Again purple and white flowers in the second generation. No light purple or pink flowers were formed. So characters were not mixed. We did not get any intermediate characters. So in both generations, all the plants were with the features of parent generation, that is P generation. No pea plants were with intermediate characters. All these were observed. Then Mendel assumed that it was because two different factors determine a single characteristic. That is very important. He said two different factors determine a single characteristic. That is this. Now when we say height of plant, tall is one character, short is one character. So he assumed two different factors. So these are the factors. These two factors, tall factor, short factor. And that determines the height of plant. That is the characteristic. characteristic. Height of plant is the characteristic. Tall and short are the factors. Then if you take color of flowers, that is the characteristic. Purple and white are the factors. The factor responsible for purple color, factor responsible for white color. Or factor responsible for tall plants, factor responsible for short plants. So, two factors determine each character. In genetics, symbols are used to denote those factors. So, I will explain that also here. Again, we will be discussing it in the other slides as well. So, here, if it is in genetics, what we do is, we consider the first letter of the dominant character to represent the factor. So, tall character will be given the symbol capital T. Whereas for the short character, we don't use S. Although the first letter of short is S, we don't use S. Instead, we use simple T. Simple letter of the first letter of dominant character. So, capital letter of the tall dominant character as the letter there. So, this is capital letter and then simple letter. So, if it is round and wrinkle seed. Now, you all know round is the dominant character. So, we use R, capital R for round seeds. But for wrinkle seeds, you don't use W. Instead, you use simple R. So, like this. If you consider the color of flowers, that is the characteristic. What are the two factors? Purple flower and white flowers. And out of these, what is the dominant character? You can remember in the F1 generation, it's only purple flowers. F2, purple is to white, 3 is to 1. So, purple flower is the dominant character. P is the first letter of purple. So, we write the capital P for dominant character purple flowers. Whereas we use the simple P for white color flowers. Remember, you don't put W. You use capital P, simple P. Capital R, simple R. Capital T, simple T. Now, another example. If we take, say, yellow and green seeds. Yellow and green seeds. Yellow is the dominant character. So, what do? We use capital Y for yellow seeds and simple Y for green seeds. We don't use G. You have to remember that. So, likewise, if we go back to the previous slide, here you can see seed color yellow, round. Then if it is for the shape of the pod, what will you do? Inflated is the dominant character. So, you will use capital I for inflated character, simple I for constricted character. Color of the pod again we discussed, that is a different one, green and yellow. So, here green is the dominant character for color of pod. So, they are capital G and simple G for yellow. So, this is something you have to understand, students. Here color of the seed, yellow and green, yellow is dominant. Color of pod, green and yellow, green is dominant. So, if it is axial and terminal flowers, capital A, simple A. Tall and short, of course, capital T and simple T. That is how we use the letters. So tall and short plants, round and wrinkle, this is for the seeds. Then here yellow and green is for the, again, seeds. Purple and white for the flowers. Purple and white flowers. So just for you all to understand, yellow and green, just to again confirm, here you can see yellow and green seeds. Yellow is the dominant color, so we use white. If it is pods, green is dominant. So if we write that also, green and yellow pods. So then, of course, we use capital G for green, simple G for yellow. You have to remember. Because students, they can give you all any of these characteristics and they can ask you to explain the observation and how characteristics are inherited from generation to generation using the letters and explaining it in genetics. How we do the explanation in genetics. So for that, you need to use the letters. Is that clear to you all? So if I go back, here you can see in genetics, symbols are used to denote those factors. And now we will see how we can denote the symbols that we have already done. We will now see how we can use these symbols to explain the characteristics. So the standard is that the dominant factor is denoted by a capital letter and the recessive by a simple letter. That is what we discussed in the previous slide. So here, if you look at this, you can see accordingly for tall feature. Tall is the dominant character. So what was the letter we used? The first letter of tall, capital T. And for short feature, we use the simple letter of the dominant character. So since this is capital T, short will be simple T. So from that itself, you will be able to understand what is dominant and what is recessive. Then, for each inherited feature, two factors are involved. Now, according to Mendel, two factors determine the feature. Now, there are purebred plants and plants with mixed characters which show the dominant feature, where the dominant feature is expressed. So, if both characters are same, then it is the purebred plant. So, here pure breeding tall plants, in that since it is a pure breed, both characters are tall characters. So, there you need to write both the factors, two factors. We will be writing it as capital T, capital T. Why pure breeding tall plants? So, both have to be tall characters. The two factors have to be the same tall character, tall factor. Then pure breeding short plants. Again pure breeding. So both the factors have to be the same. Here it is the short plant. So we will be writing it as simple T, simple T. So pure breeding tall plants, capital T, capital T. Both factors are same and it is the dominant character. So capital T, capital T. Pure breeding short plants, both factors are same, simple T, simple T. Simple because it is the recessive character. You have to remember that. Then there is another plant where it is not a pure breed, but these are the tall plants with recessive short feature. They are not pure breeds. So there is a mixing of characteristics. So the factors will be capital T and simple T. But since the tall character, capital T is the dominant character, simple T, that is the short character, is the recessive character. The plants will be tall plants. That you have to understand. This is the tall plant with recessive short feature. Although the factors are mixed, tall and short, still the plant will be tall because tall is the dominant character. You all have to understand that. So here you can see this is a tall plant, both capital T, so it is the pure breed plant. This is also a tall plant, but there is a recessive character as well. So it is not a pure breed plant. Always short plants are pure breed plants. Why? Because short characteristic is the recessive characteristics. So always when there are both recessive factors only, the recessive character will be expressed. That is why you will understand again when we go through the Mendel's observation. That is why in the first generation there are no short plants. Only when there are both factors recessive, the recessive character will be expressed. And that is why we get the ratio 3:1. I will explain that again when we observe or when we write and explain the inheritance of characteristics. So then, when two factors are similar, they are known as homozygous. I will explain this to you all. When the factors are not similar, they are known as heterozygous. So, homo means similar, hetero means different, zygous means these factors. Now, if you take these two, both factors are tall. So, both factors are similar. If you look at this, both factors correspond to short. So, both factors are similar. So, these two, pure breed tall plants, pure breed short plants, the factors, they are similar. Tall, tall, short, short. So, both these are known as homozygous. These two, they are known as homozygous. Whereas the tall plant with recessive short feature. Here there is a tall feature capital T, short feature simple t. That is what is known as heterozygous. Heterozygous. So this is heterozygous. Do you all understand that? Now the same way, if we write for another character. Let's say color of flower. Flower. If you look at the table, the observations of Mendel experiment, you will see purple is the dominant color. So, what is the letter we write for purple? We will be writing. I will first write down the factors, then we will write down the letters. Then we have white flower. White is the recessive character. So, for pure breed plants with purple flowers, pure breed plants with white flowers, then plants with purple flowers that contain recessive factor. So those are the types we discussed. Now then, if we write the letters. Now for color of flower, purple color is dominant. So how do we write the letter? Purple, dominant means capital P. White color is recessive. So we use simple P. Capital P, simple P. Then pure breed plants with purple flowers. Both are pure breed, capital P, capital P. Pure breed plants with white flowers, it will be simple P, simple P. Now both these, what do we call them? Both these are homozygous. Homozygous. Then we have plants with purple flowers that contain recessive factor. How do we write that? We write it as capital P, simple P. Dominant character purple flower, recessive character white color flower. Capital P, simple P. And what is this known as? Heterozygous. This is heterozygous. Heterozygous. Now do you all understand how we use letters? Dominant character, recessive character. Pure breed plants with dominant character, pure breed plants with recessive character, a plant with the dominant character, but there is a presence of recessive factor. Homozygous, heterozygous. Like this, I am sure you all can write for round seeds and wrinkled seeds. Can you all try that? Once you try that, you can check that with my answer. If we do the next one. Shape of seed. Shape of seed. Round is dominant. Wrinkled is recessive. Then we have pure breed round seeds. Here I am writing it short again. Pure breed plants with round seeds. Pure breed wrinkle seeds. And round seeds with recessive factor. So how will you write the letters? Round, dominant character. So we use capital R. Wrinkle seed, recessive character, we use the simple R. If it is pure breed plant with round seeds, it has to be both capital R, capital R. Pure breed wrinkle seeds, it will be simple R, simple R. Then round seeds but with recessive factors, it will be capital R, simple R. When both factors are similar, what do we call it? Homozygous. So these two, homozygous. And this is heterozygous. Homozygous, heterozygous. Is that clear? So far students, we have discussed how to represent the factors that determine the characteristics. So this is in genetics. Now Gregor Mendel did not know about these genetical symbols. So in genetics, we use letters to represent factors and characters. Dominant, recessive, homozygous, heterozygous, and always the characters are determined by two factors. If both factors are similar, homozygous. Normally the pure plants, pure breeds have similar factors, either dominant or recessive. But if there is a mixing of character, presence of a recessive factor, then it is not a pure breed plant, it is a heterozygous plant. So there capital R and simple R, capital and simple letters are mixed there, because the dominant and recessive factors are mixed there. So using these symbols, using these letters, we can show and explain how characters are inherited from parents to F1 generation, from F1 generation to F2 generation. That is what we will discuss after this. So the monohybrid cross of pea plant, considering tall and short character, can be expressed using symbols as below. So this is what I told you all, how we can express the monohybrid cross that was observed by Mendel using the symbols. So if it is tall and short character, now you all know tall is dominant, capital T, short is recessive, simple T. And because it's two factors, pure breed plants, tall plants are denoted by TT, capital T, capital T. Short plants, pure breed short plants are denoted by simple T, simple T. So those are the parent generation. So here, we have P. I'll redo that. We have P generation. That is the parent generation. So we have the parent generation. When we say parent generation, it is the pure breed tall plant and the pure breed short plant. So pure breed tall plant, capital T, capital T. So there is a cross between tall plants and pure breed short plants. Simple T, simple T. So when there is breeding taking place, gametes have to be formed. Now you all can remember students, we discussed about mitosis and meiosis. Now during the formation of gamete, it is the meiosis process. There what happens? The number of chromosomes become half. From the diploid cell, you get the haploid cell. Now you can remember diploid cell is 2N. This is the 2N cell. Two factors. Capital T, capital T. This is also diploid, 2N. Simple T, simple T. So from that, when the gametes are formed, only half the number of chromosomes are obtained. So parental generation, then we get the gametes. Gametes will be only T and simple T. Now these gametes can combine together or will combine together and give rise to the first generation, what we call as first filial generation or F1 generation. F1 generation. Now to form the F1 generation, these two gametes combine together. Then we will get capital T, simple T. Gamete from this plant, gamete from this plant. When they combine, you will get a mixed character, where one is tall character, the other one is short character. So that is a heterozygous plant. But what is the character that is going to be expressed? It will be all tall plants. Why? Tall character is dominant. So when there is a tall character and a short character, tall character and a short character, since the tall character is dominant, all the plants will be tall plants. Only the tall character will be expressed, short character will not be expressed. That is why in the F1 generation, there were no short plants, only tall plants. Is that clear, student? Then, from that, what did Mendel do? He did the breeding for the second generation. So, here also there are gametes formed. Gametes produced. Now, for the gametes to be produced, now here see, tall and tall is the character that is seen in the F1 generation. But between F1 generation also, for reproduction to occur, to gametes to form, and the F2 generation to form, you need to have two parental plants, two parents. So there, what are the two parents? Both have heterozygous, capital T, simple T. So from one parent, the gametes will be capital T, simple T. Likewise, we need to have another set of factors. So there again, capital T, simple T. Then there will be a combination of these factors that will give rise to the F2 generation. We will have the F2 generation. Now in this, always students, when you combine factors from one flower, factors from the other flower. Actually, from the first generation, gametes to second generation, it's self-pollination, but still one is from the pollen, the other one is from the ovule. So anyway, you will have two sets of characters, two characters mixing together. So in this, the factor responsible for the tall character can combine with the factor responsible for the other tall character. So I will use a different color. This will combine with this. So here, for you all to understand, I am using different colors. But when you all write, you use one color. So here, these two can combine. The capital T combines with capital T. So both are dominant characters, tall character. This plant will be a tall plant. Then, this tall factor will combine with the short factor from the other one. So there what will happen? Tall, short. This is homozygous, tall plant. This is tall and short, heterozygous, but still the plant will be tall. Why? Because tall is the dominant character, short is the recessive character. So this is also going to be a tall plant. Then we have this factor, short factor, combining with the tall factor there. I will use another color, like I said, then it's easier for you all to understand. This combining with this one. So you will get, although simple T and capital T, when we write, we have to write the capital letter first. Then only we write the simple letter. So capital T, simple T there. Then again, the last two factors, simple T and simple T. Now you look at all the combinations. Capital T, homozygous, tall plants. Capital T, simple T, simple T. Those are heterozygous combinations, but still they will be tall plants. So all these are going to be tall plants. Only this one, simple T, that will be short plants. Is that clear? This is short plants. Tall plants, short plants. Is that clear to you all, students? So you have the homozygous tall plants, heterozygous tall plants, and homozygous short plants. That is how we get the F2 generation. So here in the F2 generation, students, you all can see 1, 2, 3. 3 combinations give rise to the tall plant, that is the dominant character. Only one combination, the homozygous recessive characters, recessive factors, give rise to the recessive feature. That is the short plant. So can you see the ratio? 3:1. 3 tall plants, only 1 short plant. So that is the ratio observed by Mendel. Now if we write that down. Now this ratio, tall is to short, is known as the phenotypic ratio. Phenotype ratio. Phenotype ratio. Phenotype ratio, that is tall is to short. And that is going to be 3:1. That is the ratio of the second generation. Tall is to short. 3:1. This is known as the phenotype ratio. Phenotype means the characters that are expressed. Tall character, short character. And here, these factors, capital T, the homozygous tall character. Capital T, simple T, heterozygous tall character. Then simple T, homozygous short character. Those factors responsible for these characters are known as genotypes. We will be discussing genes when we discuss the concept of genes, but here you will have to remember they are known as genotype ratio. Now when we say genotype ratio, capital T is to capital T, capital T is to capital T simple t, that is to simple t. What is the ratio? You have 1 capital T, 1. But you have capital T, simple t combination 2. So here it is 2. And that is to simple t 1. So this is known as the genotype ratio. You all have to remember these. When there is a monohybrid cross, you get the phenotype ratio and genotype ratio for the second generation. Even for the first generation it is there, but there is no ratio. Why? Because all the plants are tall plants. So the genotype is going to be capital T, simple t. And the phenotype is going to be all tall plants. Only for the second generation, we have a ratio. Tall is to short is 3:1, but genotype ratio capital T is to capital T, simple t is to simple t, 1:2:1. That is how we get the ratio. I'm sure you all can understand this. So that is how we explain how characteristics are inherited. So here you can see the monohybrid cross of pea plants considering tall and short character. Parental generation, tall plants, pure breed, short plants, pure breed. From that you get the gametes, tall gamete, short gamete. And F1 generation is a mixture of both tall and short character. It is a heterozygous tall plant. So if we get the gametes from the first generation, tall and short gametes. And when they combine, you get the F2 generation where 3 plants are tall, only 1 plant is short. So from that, we get the phenotype ratio, that is the character that is expressed, tall and short. Then genotype ratio, TT is to capital T simple t and simple t is to simple t, that is 1:2:1. That is how you get the genotype ratio. And this is how characteristics are inherited according to Mendel's observation. I'm sure you all can understand that, student. So with that, I'm going to end this chapter. But in the next chapter also, I will be discussing a similar inheritance with another character, and after that I will explain to you all how the same thing can be represented using a Punnet's square.
To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.